This episode of In Vivo is going to change your perspective on something that is a nightmare scenario for most. And that's the idea of something larger than bacteria living in your gut. Or in other words, gut worms. But for Dr. Andrea Choe, millions of years of coevolution offers an opportunity for an entirely novel approach to treating autoimmune diseases. Dr. Choe's journey begins with a hypothesis that might sound counterintuitive at first. The idea that gut worms, often considered harmful parasites, have co-evolved with humans in a way that fosters a unique kind of symbiosis. This theory is rooted in millions of years of shared evolutionary history. Evidence from ancient remains like those of Utsi the Iceman and King Richard III reveals the historical presence of gut worms in human hosts, highlighting their deep biological connection with us. And what makes these worms so fascinating is their ability to secrete small molecules that interact with our immune systems. Far from being mere invaders, these molecules appear to have immunomodulatory effects, subtly influencing our body's inflammatory responses. In regions where gut worms are still prevalent, there's a notable reduction in autoimmune diseases, suggesting that these creatures might hold the key to rebalancing overactive immune systems. This is where Holoclara comes in. Dr. Choe and her team are working to synthesize these small molecules in the lab, bypassing the need for live worms while retaining their beneficial properties. Their mission? To harness this ancient partnership to develop safe and effective treatments of allergic and autoimmune disorders. Holoclara is already making history with their first ever worm-derived molecule entering phase 1 human trials. In today's conversation, you'll hear about the science behind this innovative approach, the challenges of translating it into medicine, and the broader implications for how we view our relationship with nature. Join me in vivo with the CEO of Holoclara, MD PhD, Dr. Andrea Choe. Dr. Choe, as of today, you and your company Holoclara are officially the first to have a worm-derived molecule in phase 1 human trials. Congratulations. How does it feel to see your vision come along so far?
I'm just so grateful. I think I'm very proud of our team. It takes a village, and it's everyone's dream when you're in the medical setting or the research setting or an MD PhD to go from bench top to bedside, they call it.
Right.
So having that realization that we're at that moment is really incredible.
Now, before we delve into the idea of immunomodulation and gutworm symbiosis, take me back to where it all started. Because, for most people, when they think about gutworm infestations or just gutworms in general, it sort of brings up images of horror movies or even sci-fi, but you have a very different perspective. So what got you started on that road to bring you to where you are now?
Yeah, well, they're cute. Have you seen first of all, I started in a C. elegans lab, right? So have you seen what C. elegans looks like?
I have, definitely.
Right? They're cute.
You're right. They are cute. That's for sure.
Cuties.
Very cute. And ironically, you're actually the third person back to back that I've interviewed on worms. So I've got a nice big shot of worm science over the last couple of weeks. So become familiar with that. So I can see that. It's cute, definitely, for sure.
Yeah.
So you have a very interesting story about how you came to study or get involved in a lab that was looking at worms. Your PI went on vacation and you decided to sort of change your trajectory. Tell me about that.
Yes, he went on sabbatical, as a lot of Caltech professors do. They're called to teach elsewhere. Woods Hole is a popular place or another NIH site. And Paul really did do that. He helped to bring his genetics expertise across the country. So when he left, I think that most times mentors are there when you're wanting to veer from your original idea of what you want to do, to make sure that it's because you're doing it for a good reason. It's not that you're losing faith or losing hope. So, I didn't have him there to turn me back towards the original project. And I'm glad, because I was just so interested in understanding how worms communicate with each other. It was just so compelling. And actually, looking back, he was happy that I went rogue. To this day, I think he has a twinkle in his eye when he talks about that.
Well, as I understand, he's part of Holoclara, is he not?
Yes, he's a co-founder of Holoclara.
So he couldn't have been that upset when all was said and done at the end of the day. So
Right.
You have this very interesting thesis that's taken you years to sort of develop into where you are now. Your first sort of, like I said, the first gut or worm-derived medicine that's now in phase 1 trials in humans. And the idea is that, as I understand it, is that there are millions of years of coevolution between gutworms and humans. And in fact, there's even a very interesting historical example where the Iceman in King Richard III had the exact sort of same gutworms living in their system. So they've definitely been around for a long time, and there is this symbiosis that's involved. So what exactly is your hypothesis? And because you're also a PhD MD, so you have some real bedside evidence that you saw when it came to gutworm presence and autoimmune disorders like asthma and that sort of thing. So what is the hypothesis, and how did it lead you to where you are now?
Yeah, thanks for asking. I think that those examples that you gave are great ones, right? Because you see Otzi, is that how you pronounce it? Or is it Otzi, the Iceman?
Utsi,
Right?
yeah.
Utsi, is it?
Yeah, I think so.
There's Hola somewhere. And
Yeah, that's right.
the oldest European natural human mummy found, I think, somewhere like Austria, Italy, right?
Yeah, in the Alps somewhere.
Yes, and that was from 5,000 years ago. And so you see that he had gut-dwelling roundworms. He had whipworms, if I'm correct. And that's not how he died. He died from the arrowhead that was found wedged in his shoulder. And then you look at King Richard III. It's not. People have this misconception that people that are living in very rural areas, right, are the only ones to have roundworms or gut-dwelling worms. And even the king of England had gut-dwelling worms. So it was pretty widespread, and it has been with us as we were our early human ancestors through the time of King Richard III and Otzi, the Iceman, and to modern day, probably up until the 1900s, when that's when it was more common to get rid of them because we thought they were gross. So I think that if I were to speak about the significance of that, the significance would be that, we see them as foreign invaders, but could it be that they are actually part of our human biology and that they always have been up until the time that we removed them? So that's the new perspective that I would bring in. And where did you go from there? Where you were asking from those mummies? What is
the well, there's also some other evidence that's sort of just to back that up, is that Africa has the highest incidence of roundworms in the gut, but the lowest incidence of autoimmune disorders. So there is definitely something going on there. That I don't know if you call that anecdotal, but there's that evidence there. And I guess my next question for you is, why is that? There's obviously time has gone by, an incredible amount of time, and these worms are still around, although we sort of forced them out. Why are they still there? What do they get from us? And then the more important question when it comes to Holoclara is, what do we get from them?
Mm-hmm. Well, that's their home, right? So if you are a gut-dwelling worm, your home is the gut. So then you rely upon a mammalian host. And I think that your eggs can be dormant for quite a long time outside of the host. It requires the host to thrive and grow into adulthood.
To complete its life cycle, essentially.
I think it's fitness. It's evolutionary fitness. It's their desire to survive, which they've done because they've been here since before humans, right?
Mm-hmm.
Lucy, the Australopithecus was 5 million years ago, or something like that. And then before woolly mammoth, 10 million years ago. And then dinosaurs, 100 million years ago to the era where there was nothing but like ferns and trilobites, 500 million years ago. And the worms have been here and succeeded. It's survival. It's their desire for survival. I think where that could benefit us is that, I think that I can't speak for all worms. I don't know all worms, right? I think that there are some that can live very symbiotically with us. And, their interest is to live in a happy home. So, over time, they have had the luxury of time to try to produce a whole toolkit of molecules to help to improve its host and its home. So I think if we can harness those specific molecules then we can return that back to the human body and think about how we could use that for our benefit.
Right. And I mean, I've heard you mention, and I've read stories of, sort of this, whenever there's a technology or a medicine, there's always some sort of black market, if you want to call it that develops. And there's a whole population of people that are purposefully infecting themselves with roundworms because they're seeing some sort of effect when it comes to their autoimmune disorders, right? And that's not something that I know, that's not what Holoclara is doing, or maybe, you may or may not be advocating for that, but it's something that people are doing, right? And which seems odd. But the idea of, I don't see a big leap from the idea of the microbiome, right? Which is something that people are very okay with nowadays. In fact, they'll go to the grocery store and buy a shot of some sort of probiotic, prebiotic to help that happen, right? Now, maybe you want to call it the macrobiome in the gut, where you're talking about larger than single cell organisms living in the gut. So that I don't think it's that big of a jump. Maybe for a scientist, it's not that big of a jump, but for lay people, it might be a big jump. So the idea of deriving these molecules is such a great idea. And I think it's awesome that you guys are doing that. Now, what is it exactly that you see? I guess the term is immunomodulation, right? This is what worms are doing in terms of how we're benefiting from that. So what is your hypothesis there? And what sort of evidence have you seen to suggest that having worms within you and then extrapolating that to molecules is of benefit to us?
Sure. And then just to comment on something you said a minute ago, I think it's totally fine for someone, if they want to embark on a journey of taking a worm, then Godspeed, because every person is entitled to make that decision for themselves, a risk and benefit ratio.
Absolutely. Right? And there are stranger forms of treatment, that's for sure.
For sure. And there are always medications to get rid of worms. So in that way, you can try it. And if you don't like it, you can also take those medications to get rid of worms. I've talked to people that have taken worms. I have talked to people that have given their children worms, and they've seen remarkable recovery. So I think that that's totally one way to go. It's almost comparable to taking a probiotic, because the species of probiotic will tell you lactobacillus, whatever, right? Akkermansia, whatever. It's the same thing as taking a Trichuris, whatever, right? Yeah. So I just think that it's a matter of perspective. And just like you said, it's not as big of a leap to think about something from the macrobiome perspective. It's just nobody likes to think about that.
Right.
And the beneficial part, I think that there's tons of evidence showing that there's benefits to having a roundworm. There's this really incredible paper was published in Nature that studied this tribe of Bolivian women, and the ones that had a particular species, I think it was Astrus lumbricoides, they had more children at an earlier age. They were more fertile. And I think that was an Aaron Blackwell study. It was just so interesting to think about why. And it makes sense from the perspective that if you're a worm and your body and the host is now sensing a foreign agent, and then it's trying to get rid of it, you can also have off-target effects. You're starting to, try to get rid of a lot of things in the body. You might not be targeting exactly that worm. So the worm has probably evolved something that can help with immunomodulation so that it can live quietly in the host. And so when you have a fetus, your body also can think, oh my gosh, there's something foreign in my body. Let me send something to get rid of it. The worm is also secreting something that's also helping to modulate that. Say not so fast. There's no reason to send the bazookas, right? So I think that that's an example of a benefit that's been seen in literature. And there's countless other studies out there that in a controlled laboratory environment, that was a human study. So now if we go to controlled laboratory environments, there are academics all over the world where they have a mouse model of disease, they return the worm, and then they see that there is a protective effect from obesity, arthritis, allergic and autoimmune disease, even to possibilities of longevity. So I think that there's just already a lot of evidence out there.
So there's an interesting serendipity. I don't know if it's serendipity or would you call it, is it the chicken or the egg? But it is an interesting situation where you have the worms that would otherwise be kicked out if you weren't able to knock down some of the effects of the immune system, hence the term immunomodulation, turning it down. And then for anybody who's predisposed to sort of autoimmune disease, which for the most part you can define as too much inflammation going on somewhere. So that's being turned down. So it's which one came first, I don't know, but it's a fascinating symbiosis that's occurring there that you guys are trying to take advantage of. Now, I wanted to ask you, when people think about bioprospecting or pharmaceutical discovery, most of the time they think about things like the rainforest. It's often quoted as the rainforest is the greatest source of molecules and potential medicine out there, right? They don't often think about worms. Nobody's really thinking about worms other than you and your small group of scientists that are in that world. How is bioprospecting in worms different than bioprospecting, for example, in the rainforest? Practically speaking, I guess.
Yeah, I think that the rainforest, why is it such an interesting place for people to go for that bioprospecting, you're calling it?
Mm-hmm.
Probably for the diversity. Is that your thinking as well? Because
I think it's the diversity, but there's also a inherent ancestral knowledge that's there, right? You've got depends who you ask, 10, 20, 30, 40, 100,000 years worth of indigenous knowledge there, hints as to what could be used for what. So I think that helps center or guide scientists in a way. But when it comes to worms, it's kind of a blank slate. It took a lot of time for somebody to realize that. So I think that's why people tend to go towards the rainforest. But when it comes to the worms, now you're bioprospecting or mining for molecules in worms. How does that actually work? How do you focus in on one molecule, right? Because you've got worms that are secreting signaling molecules for quorum sensing and that sort of thing. How are you honing in on the one molecule, for example, that you've got in phase 1 trials right now?
Yeah, I would say, to go back to your rainforest question, I think that if we have evidence that there's something in the rainforest that, first of all, may have co-evolved with human biology, or there's a tribe living in a specific rainforest where they're eating something specific and you see that they're having this therapeutic effect, then I would for sure go there. I would love that. Rainforest medicine sounds lovely to me. And so I think that with worms, we're looking at the ones that we think have that effect, or we know actually because of the pre-existing literature. So it's all the work of the incredible people that have come before us. There's a lot of great worm biologists out there that have already done this, where they really explored what are the types of species that can do these sorts of interesting things. It's just no one's bothered to pick up the baton to go further. Now let's study those species and see what they're making.
Is that because there's something inherently difficult in mining in worms? Why hasn't anybody done it up until now?
Well, there's two different ways to think about it. One is mining the small molecule space, and one is mining the proteomic space. And I think in the proteomic space, there has been efforts, and it's been challenging. I think that with proteins are much easier to find. There's a finite universe of that. Right. And there's 500 amino acids total in the world, right, that we share amongst all living creatures. But small molecules, that's not the case. It's infinite dark matter of space. So that's where I'm naturally drawn. And I think in terms of why no one's done it before, I would say that maybe no one wanted to be the crazy worm lady. I mean, it seemed like a hard sell. I think when I told my friends when I first wanted to do this, like, oof, aren't you gonna be a doctor? Aren't you, like, people want to go to what's known for types of careers that make sense. Right?
Sure, the path of least resistance, right?
Path of least resistance, path of comfort. Yeah. And I think that trying to open up with worms can be off-putting for the reason that you opened with this podcast. So people often think of the sci-fi version. But if you've sat in a roundworm laboratory and you're holding them in a petri dish and you're looking at them, you might think otherwise. So maybe I had the benefit of that initial experience where then I didn't feel grossed out by them. It felt very manageable to work with them. And I just didn't have that knee-jerk reaction. But yeah, anyway,
I wonder if you agree. I wonder if you agree. I feel like there's something that you can tell about a scientist and the model organism in which they've chosen to work. Right? You've got mouse model people, you've got tissue culture people, you've got worm people, fly people, right? And I think I've met a couple of worm people now, and they all seem. Stop me if you agree or disagree, but they all seem very. How should I say? They love the hard path. They don't like the path of least resistance. They like a challenging thing. They like to discover. They like the novelty of it, or the unknown, I should say. There's a lot of unknown. There's lots of known in worms as well. Like, for example, C. elegans. We know every single cell that exists in C. elegans, as well as all the neuronal connections. So we know a lot, but there's lots of unknown as well. And with worm people, I think that I've seen that trend. I wonder if you agree or disagree.
I think so. So I've been all of those. I was in a fly lab, I was in a mouse lab, and all of the above. And I like all of those peoples. And I think that if I had to look back with worm people, there is a huge sense of community, of sharing. There's a worm consortium where people gather worms and send them out if you request it. Anytime I've asked someone for a worm, they've kindly sent it, sometimes with a handwritten note. There's always whimsy. I think that I remember there's the twinkle in Sydney Brenner's eyes is notable. But Seymour Benzer had the same twinkle, and he's a fly biologist. So I don't know. Maybe maybe the microorganisms or macroorganism people, I'd say the fly folk and the worm folk, were definitely a lot of fun to be around.
I just want to go back to something you said. It just occurred to me. You delineated between, when I was asking you about, are there any inherent challenges in working with worms? You delineated between the protein space and the small molecule space, right? And that's an interesting delineation, because when it comes to proteins, they're difficult to work with when it comes to actual treatments and molecules, right? It's something you can't take orally. Most people would prefer to avoid injections, and that's because proteins get broken down in the gut. And so, human growth hormone has to be injected, as an example, or insulin has to be injected. But small molecules are different. You can take them orally if they're developed the right way. Is that one of the reasons why you decided to go that way?
For sure. I think that proteins are easier to find up front and get to clinic, but then you have challenges afterwards for the reasons that you've said. And also, you can have an immune response to the protein. Yes. Right? Now your body is detecting a foreign protein. So then the next time you take your treatment, you can have an immune reaction to that. So then it becomes muddy, right? You're not getting as strong as an effect and might have an adverse effect down the road. I think small molecules are much more challenging up front. And once you've identified them, and if you can definitely develop it to be orally available, then you have much more to work with, not just in the fact that you can make it orally available. You can make it from chemistry. You're not growing batches now. You're not secreting things out of batches. You're not asking what was wrong with batch A and batch B. Right. Why did cells secrete it this time or that? What trying to deconvolute that can be quite complicated. But also, you can make modifications as chemists to your molecule.
Right.
So then you can say, hey, what can I add to my molecule to help it recapitulate what a worm is? A worm is secreting this in a gut. We're not implanting something to secrete it out of the gut. We want something traditional where people can take it and it leaves their system. Oh, is there any modification that we can make that can make it more permeable to the gut, or last longer? So we can make those simple modifications. So as long as you have great chemists around you. So I think that there was more tinkering up front too that I liked.
So you answered my next question, which was going to be: are you expressing these molecules in a host organism? Are you synthesizing them? Or are you extracting them? So you are synthesizing them.
we're synthesizing them. We start with the worm, we find the thing that we want, we make it from scratch, we make sure that it's the same effect that we are looking for. Then we then synthesize them from scratch. We ask, can we make this better? Let's try that out. Then you pick your lead out of that group, and then you move forward. So
Now,
we're very much leaving behind this idea of growing them in vats of worms or however.
Right.
That
I mean, when it comes to biology, there's so many variabilities involved. Like you said, batch to batch, it can be a nightmare if you're dealing with fermentation or bioreactors in large volumes. And like you said, one batch is different from the other. I can imagine that would be a nightmare when it comes to actually making a product. Small scale, benchtop in a lab, a lot easier to handle, but scaling it up to that level would be, I would imagine, a nightmare. I've never been involved in any pharmaceutical discovery, but I would imagine. Now, one thing I heard you mention in a talk with the Aspen Institute, which I thought was fascinating, was the way you outlined your approach is that you're trying to keep things as close to Mother Nature as possible. And I thought that was very refreshing. When you hear about pharmaceutical companies, I think that most people sort of have this very sterile idea as to what you're dealing with. Stainless steel, clean rooms, and which is probably still the case in what you're doing. But your approach was very refreshing. Can you talk a little bit about that?
Sure. I think that on one level, it's just because I've been humbled by my own ego. Anytime I've thought about trying to make something better, I usually don't. Right. I tend to make it could be worse than what was better. And I don't know, sometimes I even think about it like the first time you take a picture and then you're like, no, I can do better. And then you take 15 more and it's always that first one that's the best. Right. So I think and it's also what is man-made. I just think that we have not taken into account. How can we know as much as what the worms have had the benefit of millions of years, right? Not just millions of years, but hundreds of millions of years. Me with my small brain cannot beat that.
That's right.
That if nature has innovated something, we have really better take a hard look at that and make sure that that's if we're going to try to beat that or iterate on that, which we have, by the way. We have thought about how we can improve it. But I always favor what Mother Nature has innovated because there's a reason that it contributed to fitness and it survived and it's around. So I just think that we cannot ignore that.
Yeah, I mean, there's no way you could compete with the forcing functions of evolution over millions of years. And you're right; it's sort of I think there's an ego involved in. I mean, scientists are known to have egos, but I think to assume that you can make something better than Mother Nature, I think there's a little bit too much ego involved there, a little bit too much hubris. So I love that. I love that approach.
Oh, like the Aspen talk. I remember that now. The moment that I talked to it was the Jurassic Park analogy. Yes, it was a caution.
Yes, that was a great analogy. One day
I'm going to meet Jeff Goldblum and tell him about how impactful that his role was in Jurassic Park, because I just remember looking at his face in that movie and him like his glasses on, just warning them, don't mess with nature. Nature will find a way. And of
nature
course,
always finds a way. Yes,
they didn't. They just ignored him. They didn't believe him. They plowed forward. They're making velociraptors, right? I mean, it's just that felt a ping of truth to me, a very strong ping of truth.
Yeah.
That not only should we honor what just from the amount of time that nature's had to innovate, that we should go there first, but also that very deep cautionary tale of think twice before you think that you can do better.
So you're in the West Coast, right, in California. So you're surrounded by lots of biotech and pharmaceutical startups and that sort of thing. How has your hypothesis and your approach been received in that community? Are people excited? Are they? I mean, I'm sure there might be some jealousy involved, but how has the actual thesis been received there? Because it's a very different approach. I mean, that's sort of the nature of the talk that we're having here today. It's very different and unique and novel. How's it been received out there?
I think that I've always found my supporters. And if there's someone that's not a supporter, I wouldn't know because they've ghosted me by that point, right? They're
Right.
not they're not in my periphery. So I've just always focused on who are my supporters, and they're still here with me today to this day. And if we're talking about the LA ecosystem, I went to UCLA, USC, and Caltech. So, I have connections to a lot of great thinkers and a lot of great minds that, again, have always been so supportive of this. And I would say that at Caltech, there's been a lot of interest in how can we do something new that's not iterating on a known target, right?
Mhm.
Not another GLP-1 agonist, not an iteration.
Right.
Well, what's new? And when I come to people in that ecosystem where I think about how can we think about something new and totally outside the box, it's not been scary. I think it's always been met with curiosity and further thinking and just longer conversation, hours and hours of conversation. So I just think that I've always focused on finding my people and keeping them around me.
So I'm not sure if I think you mentioned it, but you are a rare breed in that you are a PhD, an MD, and now you're an entrepreneur. So you sort of are best of all worlds when it comes to biotech and pharmacy. You've got a lot of perspective and a lot of, you've seen this sort of world from different angles, all three angles. What do you consider yourself most at this point? Are you entrepreneur first or PhD or MD first? How do you look at it?
Such a good question. And I think that day to day, entrepreneur is really at the forefront of my mind. I'm always thinking about how can I be a better leader? How can I be grow, move my team forward? What's ahead of me that's in the biotech space that I could learn from? How can I surround myself with those types of folks? But I would say that my scientific mind is always probably my primary driver, the way that I ask questions, and I'm curious. And it's not just I'm curious, I'm intensely curious. So I think that that's always there, but probably ever since I was a child. And I think that the medical side is my North Star. At the end of the day, this is just about patients. How do we get to them sooner? Who can we help? Who can we help first, second, or third? What do they need? What is their feedback to their current treatment regimen? And then I would actually go and meet with them and ask them, what do you think about this? And we love the idea of something from nature. So I definitely, all three of them are drivers in different ways.
If you don't mind, can we talk about Holoclara a little bit?
Sure.
How big is it right now in terms of employees? How big of a company is it?
So we have 10 or 11 people currently at the company. And we have partners. We have a lot of collaborators and partners that work with us.
I'm asking that question specifically because you talked about I try to become a better manager. So I wanted to get a sense of what you're managing exactly. Do you look to anybody in terms of inspiration for management? Is there anybody in the tech business that you idolize or? Yeah.
I don't know that I idolize anyone, but I'm always learning from everyone. I'm learning from I learn a lot from Joe Betts LaCroix over at Retro. He works with what's his name? Fred Kaufman, right? So, learning a lot from what he learned from Fred. I have my own coaches that I work with. I learn from people on my own team as well. I have incredible people around me. I've hired people that have been in this industry for decades. I consider them my teachers. Interesting, I actually learn a lot from my own nanny who works with my son, and she really studies conscious discipline and how to really lead with questions and curiosity. And, I think that I learn from everyone in that way. I'm sorry if that's such a broad answer.
No, now I'm curious. What is conscious discipline?
Conscious discipline is the field where, the way that you interact with your child, it's not just about discipline. It's about being conscious in terms of having them be more aware and supportive of their own needs and empowering them with the tools to deal with it. So And you yourself as a parent are being more conscious about how you're disciplining your child. And so you being very aware of when you're giving off energy of because I said so, but still giving firm boundaries.
Mm-hmm.
You never sugarcoat you don't have to sugarcoat something, right? But you're being mindful of how the message is being conveyed so that they can hear it, and being aware of when they can hear it and when they cannot, right? So if my son is in a tantrum, he's in his lizard brain, I don't mean not to be disparaging, but he's in his lizard brain. So you're not going to give them feedback at that moment. You're just going to work on them having that space and that self-awareness to say, I need to take a moment and then I can hear you. So it's the same thing with people too. I'm a person that's very action oriented, but the downside of that is I can also want to just give feedback rapidly. So you have to take into consideration how and when a person can hear something.
Mm-hmm.
There's a very, very bi-directional path of growth for both the parent and the child. And in my situation, a leader and who I'm working with. So it's a lot of work, but it's actually very fulfilling to think about it as us all being on a path of growth together.
Gotcha. Are you allowed to talk about the molecule that's in phase 1 trials right now?
What do you want to know about it?
Well, I know it's for eosinophilic esophagitis, which was thought to be a rare sort of autoimmune disease that is involved, or eosinophils. These are granular cells of the immune system. What are you hoping to achieve with that molecule? I know so phase 1 trials, first of all, you're doing that in healthy people who are volunteers. So you're looking at basically whether or not there are any negative side effects in healthy people before you move to treat anybody. But when it comes time to treat somebody, what is it that you're hoping to do? And what have you seen in your data to suggest that this molecule in particular might be the one?
Yeah, that's a great question. I would say, first of all, with the phase 1 trial, it's not just about safety, but it's also about pharmacokinetics. So when with it, is it behaving the way that you think it is? When you give it, you take blood after 1 hour, 2 hours, 4 hours a day or whatnot. Is it now steadily decreasing at the rate that you would expect it to? And then from there, it can help you inform dosing, right? Because if it's gone suddenly, you may need to dose twice a day. If it's tail, then you dose once a day. And then you think about the effect of your molecule and all of that. So I think that it helps with dosing and dose selection, which is why we shared our indication at the same time as our phase 1, because that's very much on our mind. We're already preparing for those trials and think about how can we dose, how can we structure our trial in that patient population. And I would say separately, we know that our molecule works in multiple different diseases, both allergic and autoimmune. So I think why that's so interesting to me is it's not a broad immunosuppressant, but in the context of autoimmune disease, which oftentimes the players are a specific type of cell, then it can help to be productive there. In the context of an allergic disease, which can be more a Th2, T helper 2 type response, it can help to, if the T cells go up, to bring them down. But it's not broadly immunosuppressing things. So it's very context specific. So I think with that, there's great opportunity to expand into other indications. And we're starting with eosinophilic esophagitis. But from there, we'd love to be able to broaden out and treat other patient population.
Have you gathered any evidence to show any efficacy against? I want to not to disparage those who have EE, we'll call it, but something more common like diabetes. I was going to say important, but more common in terms of the population. Do you have any evidence that that might be?
Yeah, we did. So I did a diabetes mouse model myself back when I was at Caltech. The problem that I had with my mouse model, which I think was non-obese diabetic mouse, is I was worried about the effects that I was seeing if it was something that I was making up or was being made up, because that mouse model in particular is very susceptible to its environment. It's in a pathogen-free environment in the mouse vivarium. You have to suit up completely. If there is just normal outside bacteria getting in, things from your own skin, it can be cured, right? So I think that I had explored that, and then we had put a pause in that. But it's certainly back in our pipeline. I think early on, that's the reason why we didn't first go into diabetes.
Type 2, I'm assuming, right?
This one was for type 1, is the autoimmune pancreatic cell.
Right, obviously, yes.
Yeah.
Interesting. Now, actually, what you just mentioned there is really interesting because I think every researcher has been in that situation where you have some sort of effect or data in front of you, and you have to stop to think: am I seeing what I'm seeing, or am I kind of sort of willing this or wishing this into existence? Can you talk about that as a researcher, like how you overcome that, or how you, I guess, put in not barriers but checks to make sure that what you're seeing is in fact real?
You never overcome that. That should always be on the forefront of your mind. I think that we have to be extremely responsible with how we're testing things. So one thing that I've done is that I've really focused on animal models of disease where there's some sort of antigen. So then, and non-obese diabetic mouse is this genetic strain where I think it was discovered in Japan many decades ago, where that they found a strain that had, I think, maybe cataracts in size. So they thought, I wonder if it has diabetes too, and it became a mouse model. So it could be that there's a gene there that's the driver for that model that may not recapitulate what's found in humans. And the same thing with transgenic mice. You're knocking down a gene and then it's driving the disease. But I really like things that have antigen drivers where it's like you're exposing them to something, like a pollen or a dust mite. In asthma, it's like it could be chicken powder, right? So things like that, to me, seem to mimic what we as humans were exposed to antigens and then we're reacting to them. So I like that because there is a broad way of introducing something that's then driving the disease rather than caused by one gene or some genes. I love genetics, but I worry that when you start with a genetic model organism of an animal model of disease, you're then only looking at that one driver of disease.
Yes, it's very specific. Whereas I guess, how diabetes can come about many different ways. At the end of the day, it is always an autoimmune disease or disorder, but there are different pathways that it can sort of bubble up to the surface. So you're absolutely right. I wanted to ask you, in terms of the business side of things, is the regulatory world, or I guess just regulation, different or more challenging when dealing with worms? Or is it just because, it's a small molecule at the end of the day, but is it any different? Are they concerned? Would they be concerned?
The small molecule at the end of the day, and you have to move through every hurdle that the FDA puts in front of you. So we very much follow their guidance. I have someone on my board that's former chief counsel to the FDA, and he's had many decades of experience being on the boards of all of these companies. And he always tells this story to people where when something goes wrong, he asks them, well, did you ask the FDA what to do? Yes. Well, did you do it? And 10 times out of 10, no, it's because they didn't. And so the rule here is just follow the guidance. Just follow the FDA's guidance.
Yeah. Keep it simple at the end of the day. Do what they're asking of you. And in terms of, let's say, hopefully it's this first molecule that you guys are in phase 1, but how far off are we from seeing something from Holoclara on the shelves?
That's a great question. I think that it always depends on I hope that it's not more than a handful of years.
Right.
I think it always will depend on the statistics of your results when you enter into your phase 2 and your phase 3 and your pivotal, and how much you can do, whether it's step by step or to combine them. So I think it's to be determined. I think the statistics and the data will have to speak for itself. But I personally hope that it's not more than a handful of years.
I hope so too, because it's very. I think it's a breath of fresh air in terms of pharmaceuticals, and it makes complete sense, especially with your sort of Mother Nature approach. So I'm very excited about it, and I hope it works out.
Thanks.
I also wanted to ask you now, because of the perspective that you've had, let's say 3 attack vectors as a PhD, MD, and also an entrepreneur, what sort of advice would you give to young, let's say scientists or even med students, but scientists that are looking to become entrepreneurs? So I guess let me ask it this way. What skills, what skills make a good researcher? And are any of those skills also transferable to being an entrepreneur? So what makes a good entrepreneur, and are the same skills, involved in being a good researcher or scientist?
I think curiosity and ability to hear feedback are really important. I think that curiosity, one, you just have to follow your passion. Whatever it is, your white rabbit that you're following, keep following it. It doesn't matter if anyone tells you that it's like too this or too that, too small, too non-impactful, too weird. I've been told all of those things, right? I don't know. It never really stopped me. So I think, just follow whatever it is that's the white rabbit for you. It will ultimately take you somewhere very interesting. And always lead with curiosity and think about your own, what could be holding you back. That's the feedback part. Just being really self-aware and being open to feedback. You don't have to take it, but listen to hear what others can offer. You're going to go in your own hero's journey, and everyone needs those people around them. Dorothy from Wizard of Oz, right? Everyone's watching Wicked now. So, even she had her people around her. Everyone has to have a team. So I think that curiosity and ability to hear feedback are 2 really critical components.
How long has it been since you've said to yourself, I'm going to pursue this idea of worm-derived medicine, to where you are now? How long ago was that?
Gosh, maybe a decade. I don't think that, no, not a decade, because at the time I didn't even think it was going to be me. I had my own path. I was going to go be a doctor. I thought that Caltech would find a CEO. I didn't know anything about drug development. No, why would I take the helm of this? So I think that I very much believed in this and saw the data with my own eyes and believed in the implication of this from the very beginning. When I stepped into the entrepreneur seat, no, it definitely hasn't been a full decade. It was only because I felt the necessity to. And that's why I'm here today.
I've got a couple more questions for you, if you don't mind.
Yeah.
Sort of the golden, trophy at the top of the hill for medicine has always been personalized medicine. We hear this term very often, and it's not, it's come to fruition in some cases, for sure. It does exist, but it's not as broad as one might have hoped. Theranos may have had something to do with that. But how does worm-derived medicine sort of slot into the idea of personalized medicine? Is there room for the two?
For sure. I think ultimately, I would love to have what we want to build is a worm atlas of many molecules and their specific impacts and how they can be useful, not just from the literature showing us that they can block those animal models of disease, but which subtype, which group within there can they truly serve? So it's very patient driven, but also, what is the superpower of that each specific molecule in our quote unquote worm atlas? That's where I think it could play with personalized medicine, because let's say you and I both had the same disease. Let's say you and I both had, I don't know, multiple sclerosis or something like that. It may be my T cells are the ones that are overly sensitive to my myelin. And you might have, it just might be your myelin that's nonstop sending out signals. So there may be different drivers for our disease. And I think that's where personalized medicine, I hope, can get to the point that can showcase that. I think that's why so many therapeutics fail, because they go into these trials saying, we're going to treat the whole population. And then they don't, they get a subset. And here's what makes me furious is then that drug dies on the vine, because then the FDA is like, oh, no, you didn't hit the 3-pointer that you said you were going to hit. And you're like, wait a minute, but I hit that subpopulation. It worked amazing for them. Oh, well, that's also not the market that you promised your investors. You promised them the whole market. We want to 10 billion. So then there's all of these drivers for it to be thought of in this huge one pool. But we've learned so many times that's not reality. So now if we can really get to the point of personalized medicine where not only do we have the data to support that, it's also within our mentality and our perspectives, but that's how we should think about trials and trial designs. Then I would love to play with them at that level.
Yeah, that idea of a worm atlas. So if correct me if I'm wrong, the way you're describing it is sort of like a menu of molecules derived from these worms that you can pick and choose from, and sort of almost in a compounding fashion put together something that's unique for each individual patient.
Yes, that's the hope.
That's the hope. I like it. I like it a lot. There was, I guess, before we wrap up, I wanted to ask you, how do I put this? Sometimes with medicines, there are side effects, of course, but there's also ethical dilemmas. Are there any, and I can't really think of any, so that's why I'm asking, any ethical dilemmas that might come about when pursuing gut-derived medicine?
In what way? I mean, if there's like safety factors, or
not safety factors, but I guess one question: is this an expensive medicine to produce?
That's where traditional medicine makes it. It's the same cost as traditional chemistry would make in the molecule. So,
gotcha.
Yeah, it's market for that.
That's wonderful, because
there's not some strange way that you have to, again, we're not making this in giant worm vats or now putting them in bacteria with different batches, all of that, the QC required for that.
Right. So even though it's sort of an exotic idea or a source when it comes to where it's coming from, at the end of the day, it's going to be produced in sort of very common ways. And so it should be, hopefully widely accessible, because autoimmune diseases are very common, unfortunately, nowadays. So, okay. Last question. Where do you see Holoclara 10 years from now?
Talking to you about the worm atlas menu, walking through the menu and sharing how in trials, how we've seen the impact, now it's been approved, how are the patients responding, hearing their stories, getting feedback from them, and then just I'm so curious to see who are going to be the people that can march alongside and join our mission. I have so many great people around me now, and I'm just so curious at that point, who will be alongside us to really join the mission and help build the atlas, and what will be in it by that point.
That's wonderful. I hope 10 years from now we can at least, hopefully sooner than 10 years from now, we can sit down and chat again. But it's been wonderful talking to you, Dr. Choe, and I bid you the best of luck. It's super exciting, and I think it's very inspiring what you're doing here.
Thank you. It's such a pleasure to speak with you today. Thank you
very much
for inviting me.
Have a great day.